Thanks for the opportunity. When we received the invitation, it was directed to the chair of the scientific committee, and we thought, okay, here's another presentation of the famous opinion of the scientific committee of EFSA. But unfortunately, the chair cannot attend, and we thought we'd turn that into an opportunity and actually make it a duo presentation where I'll, next slide please, I'll cover maybe a bit of the general work of the scientific committee, but then indeed, listening to the number of times that the word pesticide has been used today, I think it was not a bad idea for us to suggest that the head of the pesticide unit, my colleague Professor Tarazona, join for the more important part perhaps of this discussion. With this, let's move on to the next, please. Okay, so indeed we got this now famous request for a scientific opinion by our scientific committee with three questions that the Commission asked us. Identification of endocrine disruptors, adverse effect characterization, and testing methods. We were invited to do this work with other agencies or consulting closely with other agencies. Thank you. Next one. Nothing new there, I think, in the sense that we make also the distinction between endocrine activity and endocrine disruption, and that the difference between them is then the adversity as defined in previous WHO papers. Next one. So with this endocrine activity, we say, okay, endocrine activity plus adversity, if there is a plausible link, leads to definition of endocrine disruptor. Next one. The, as already highlighted by the previous speaker, there is a reference point there for when it concerns test methods, and that's the OECD. The paper makes a number of points saying that for the classical EATS there may be test methods, but as you just heard, they're far from perfect. But that's, even with those considerations, that's mostly limited to mammals and fish. And then for whole life cycle evaluation, that's really missing for mammalians. The other element that is highlighted is the use of tests, interpretation of tests, where it states in the opinion that there is not a single test that is adequate to fulfill this definition, but it is further need for guidance on interpretation of results and, above all, testing strategies. We'll hear a little more about testing strategies because this opinion is already indeed 2 years old, and perhaps Jose will elaborate a little bit on this. Next slide, please. Now, I want to highlight that out of what you've heard today, EFSA did not wait any longer to address three key issues that are not unique to endocrine active substances, but certainly very much relevant there. That is non-monoclonic dose response work, where a consortium of 8 European agencies led by the ANSES in France is doing a review, thorough review there. The chemical mixture issue has been raised, and we're doing general work there, in particular with the JRC. There is also specific work with regard to pesticides that Jose will highlight. And there are a number of other issues, including the biological relevance, systematic literature review, uncertainty. The weight of evidence is a very important aspect, as is expert knowledge elicitation. But the three first ones are obviously key, and so we're trying to be proactive there, do preparatory work, and then eventually come out with a scientific opinion. Next one. Maybe Jose, I'll hand it over to you. Thank you.
Thank you. And regarding specifically in the area of pesticides, I will start with the methodological developments. The first one is linked to mixture toxicity, is the cumulative assessment groups. There are opinions on the panel covering similar and dissimilar mode of actions. One of the examples for thyroid has been already mentioned before. The other one is also relevant for the endocrine disruptor because cover the reproductive effects. And we are trying to cover more cumulative groups that will allow EFSA to be leading the possibility for having the cumulative assessment, at least for pesticides. The other key element in our view related to methodological developments are the use of epidemiological data in the risk assessment. We started with a scientific standard review, and following that, in February this year, we held a stakeholder event in Paris just to see how we can use better information from the epidemiological studies. And now we have two opinions working that the panel is working on. One is on the relationship between the exposure of pesticides and two specific diseases that were selected because of that review, Parkinson disease and leukemia. And we are trying to identify, using adverse outcome pathways, the potential link between pesticides, exposure, and these two diseases. The other one is more general and is trying to improve the contribution of epidemiological data in the risk assessment. We have identified a key element in the exposure part for the epidemiological studies. In many cases, you have a exposure to pesticides, yes, no, but there is no quantitative or at least semi-quantitative assessment. And we are trying through the panel to produce some recommendations to improve that. In the next slide. Previously, they were methodological developments, but obviously we have also the responsibility for assessing individual active substances in the pesticide. As you know, EFSA is producing the peer review for all the active substances to support the decisions of the Commission and the Member States on the approval. And I will start with the scientific assessment. The idea when you assess a chemical, and particularly for pesticides, is that you try to identify all the information together. Try to have a with evidence of all the different hazards that has been identified, the properties of the chemical, the environmental phase properties. Has been mentioned several times that obviously persistence of the chemical is also important, both persistence in the body as well as persistence in the environment for considering the hazard and possibility for a risk. And based on that, we have the toxicological and ecotoxicological profile of the molecule. This information should be coherent, and then is compared with the different criteria in the legislation. That is the best element for moving from science to regulatory science. We consider for the endocrine disruptor as well as for other hazard criteria like carcinogenicity, mutagenicity, reproductive effects, activity, so persistent bioaccumulation and toxicity, or persistent organic pollutants. If the chemical has those properties, and we identified this in the conclusions. And also, from the same set of information, we have the extrapolation of the hazard characterization, the potency, and then we move to the exposure assessment and the risk characterization. It's important to mention that for pesticides in Europe, we have a huge amount of data requirements. The legislation is quite strict. What we have now with the new regulation is not only a dossier presented by industry with the regulatory studies, the GLP studies, but also an assessment of all the scientific publications that has been published by the scientific community in the previous 10 years. Then we have the assessment by the member states and the consultation. And also it's quite important to mention that we have a public consultation on the draft assessment report that has been prepared by the member state. And obviously we will be happy to increase the contribution of the scientific community and all the stakeholders in this public consultation to ensure that at the moment that we need to have the conclusion, all available information has been presented and is available for EFSA in the decision, in the assessment process. Next slide. This is the kind of summary that we present for every single conclusion. You see the output of the scientific peer review on the different areas. The endocrine effects are covering mammalian toxicology for human health, and in ecotoxicology and environmental risk for environmental health. I would like to mention that the EFSA is quite transparent in the assessment of pesticides. So you can go to our webpage and you will see the summary dossier with a summary of every single study that was produced and presented by industry. We have a public consultation with the draft assessment report that was prepared by the rapporteur member state. And finally, in our conclusion, we have the EFSA conclusion, but also as background document, you have the outdated assessment by the member states and all the elements in the public consultation. Every single question, every single comment from the member states, the discussion in the expert groups, and the final assessment by EFSA. The conclusions identify data gaps. And as you can expect, that the identification of data gaps is quite relevant for the endocrine disruptor, but as we mentioned before, that we are still not covering all the relevant effects. We have also issues of concern, and the issues of concerns we are considering two different categories. We have a concern when we have observed some adverse effects that we are not able to fully identify and fully present the risk assessment on the hazard identification. And you will see in several of our conclusions we have issues not finalized regarding the endocrine mode of action for some of the effects that were observed in the studies. And also we have critical areas of concerns when we have a risk that has been identified that cannot be mitigated, and we identify as a critical area of concern. And finally, you have the list of endpoints with all the information that EFSA has verified and validated and can be used by the for the decision making, as well as for the risk assessment of the products that are in the market that is done by the member states. Next slide. In the area of the endocrine disruption, I think it's quite important to mention that the current information is already sufficient to identify many of the concerns. You can see that we have identified issues on endocrine level for several of the substances that has been published in EFSA conclusion recently. I think that one of the key elements is that we are assessing this as real effects. So what you see as apical studies, so the studies checking for real adverse effects observed in the animals, and then when we observe an effect that could be mediated through endocrine mode of action, we try to elucidate the mode of action, and in case that there is not sufficient information, the studies should be designed on individual basis for addressing the particular concern that has been identified for that particular pesticides. And finally, in summary, what you have in the EFSA conclusion is detailed case-by-case expert assessment of each of the active substances that are entering the EFSA process, which is based on the current scientific knowledge. All available information, also information from scientific literature, is assessed and checked for reliability and relevance. We have already identified potential concerns for some substances, leading to recommendations for generating additional data and for clarifying the endocrine mediating mechanisms. And finally, relevant effects have been included and considered in the risk assessment, even when it has not been triggered by the interim criteria. Thank you.